In this lesson, we're going to talk about the evolutionary history of life. And so at the top of the page, I've drawn this cladogram showing how our three domains are related to each other in terms of a common ancestor. And so as you can see at the bottom, I have LUCA written, which stands for Last Universal Common Ancestor. And then it branches to the left, having our bacteria, and then at the right we have our archaea and eukarya, which have a more recent common ancestor, which we can see here at this node. So they're more closely related to each other than the bacteria over here, which are related at LUCA. And so what we're going to do is we're going to kind of go through and track how these different domains are molecularly similar to each other, so we can kind of see how they evolved over time. So looking at this chart, we have our three domains broken out, and then we also have our kingdoms listed below. And so that's how we're going to categorize all of these. And so if we look at the first thing is cell type. And so we know that the bacteria and archaea are both prokaryotes, which means they have no organelles. They're very simple, single cellular, very basic type cells. So they're prokaryotes, and if we look up at our cladogram, that kind of makes sense because we're going to start out with the simplest form of life before we develop into something more advanced, which we see in our eukarya, which are protists, fungi, plants, and animals are all eukaryotes, which means they are multicellular and they have a bunch of organelles present within them, so they're much more complex. And so it makes sense that they evolved after the prokaryotes evolved a little bit later in time. And so the next thing that we're going to look at is some cell structures, specifically the cell walls and what they're made of. So if we look at the eubacteria, I already have this filled out, that their cell walls are made of peptidoglycan, and then the archaebacteria have a cell wall without peptidoglycan. So that's how we have a change there. And then when we get to our eukarya, we have another change in that our protists' cell walls are made of cellulose, or they can also contain chloroplasts within them. The fungi has a cell wall that's made of chitin, so we see another change there. So we would have another branch point on our cladogram if we were looking more specifically within just the eukarya rather than the domain as a whole. And then our plant kingdom, we have a cell wall, again, made of cellulose, and they also contain chloroplasts. And then finally, our animal kingdom, we have no cell wall and no chloroplasts. So if we just look at this, we can see how from left to right our table is changing and evolving, and that kind of follows our cladogram up above, how we see we're going from, kind of something basic, I guess, and then we're adding in the chloroplast, adding in different materials for the cell walls as it progresses to the right and evolves over time. So next, if we look at the number of cells, since bacteria and archaea are both prokaryotes, that means that they are both unicellular. And then once we get over here into eukarya, our protists are mostly unicellular. We have some multicellular within that category. And then if we look at the fungi, we have mostly multicellular now, developed that way, but we still also have some unicellular. And then if we look at plants, again, mostly multicellular, some single cellular again. And then finally, our animals we know are multicellular. And so again, with this characteristic, we can see, starting out with the basic, simplest form of unicellular on the left of our cladogram, and then progressing gradually to the more complex multicellular organisms in animals. And then if we look at, finally, the mode of nutrition, whether they're autotrophs or heterotrophs. Autotrophs meaning that they are capable of making their own food, and heterotrophs meaning that they aren't, they have to ingest food. They can't make it for themselves. And so the bacteria and archaea are both autotrophic or heterotrophic. They can be either. So it can be both. And then once we get to eukarya, again, protists can be autotrophic or hetero. And then the fungi, they are only heterotrophic. The plants are only autotrophic, they make their own. And then animals, obviously we know, are only heterotrophic. Animals can't make food for themselves, they have to ingest it. And so this chart fits right along with our cladogram up above, showing how the molecular similarities of things evolving over time, and that we started with a simple one-celled organism, and this was about 4 billion years ago. And so we can see how our different organisms, we started out this simple one-celled organism. We were able to evolve into much more complex multicellular organisms like animals and humans. And then we can also see how things branched over time and what these specific branch points stand for, like these derived characters at these branch points, where we switched from a cell wall of cellulose to chitin. That would be a branch point, and things like that. And we can also use anatomical features, not just molecular features like we looked at above, to more specifically break apart our lineages to classify organisms. So like once we get within the animals, we can use more anatomical, kind of physical features to categorize them and use as branch points to break things apart and see how different organisms are related to each other, or maybe not so related to each other. And so the next thing we're going to go over is the endosymbiotic theory, which shows how our eukaryotes arose from our prokaryotes. Because up above, we saw that we started out with the prokaryotes, the unicellular with no organelles, and then eventually we got to multicellular with organelles. And so this theory explains how that happened. And so we started out with a prokaryote, drawn here, and this prokaryote ingested an aerobic bacteria, and that was the first step to how this happened. So the prokaryote ingested this aerobic bacteria, and then if we go to our next step, we see that these aerobes that were ingested developed into mitochondria. And so we can see in the diagram, now we have a mitochondria that developed. And this is what we know to be animal cells. And then if we want to go further to see how the rest of our eukaryotes developed, now that we have this mitochondria, now that prokaryote ingested a cyanobacteria. And this cyanobacteria has photosynthetic pigments in it. So you can see in the diagram, this prokaryote ingests this cyanobacteria, and then in the fourth and final step, the cyanobacteria develops into chloroplasts. So we can see now we have a chloroplast and we have the mitochondria. And this is what we know to be plant cells. And so that is how we evolved from the unicellular, no organelle prokaryotes to the multicellular with organelles eukaryotes. And this is the evolutionary history of life.